block and either leaving it alone or converting it into estrogen. So aromatase activity is sensitive to temperature. That's the mechanism at play here. If the temperature is low, the aromatase activity is low, and so you're not getting much conversion, so you're mainly keeping your testosterone, and hence you would develop as a male. On the other hand, at a higher temperature, now we have a lot of aromatase activity, so a lot of estrogen being made, and then female development. So, seemingly pretty simple. So this can run into problems with some of the compounds that we have in our environment now that are not naturally occurring because they mimic the properties of estrogen. So one class of these is called the PCBs, which stands for the polychlorinated biphenyl compounds. And here are a few different examples of these. And structurally, they're fairly similar to estrogen, and they can even act as estrogen on some of the targets for that hormone. And the problem is that you only need to have these in a very, very low concentration in order to get the effects on sex determination, so the mimicking effects of estrogen. And so in the case of this, you can get an overrepresentation of female development for organisms that are sensitive to trying to use environmental cues to establish how many should we make male and how many should we make in females. There's another kind of famous example where there's this herbicide named atrazine that also affects the endocrine signaling. And here are the concentrations, 0.5 part per billion, really, really low, or 0.8 part per billion. And in the case of 0.8 part per billion now, these structures are just shriveled up and non-fertile. These are gonad structures. And in the case of 0.5 part per billion, you can see gonads that are developing sort of with some ovary-like structures, and then some more male-like structures. So it's sort of indeterminate whether it's going to be a male or female gonad that's developing here. So atrazine, the way that it works is that it's going to stimulate aromatase activity. And aromatase, as we said, is around to convert the testosterone into estrogen. So in control males, you should see some testosterone, a fairly high amount of it. If you treat these males with atrazine experimentally to figure out what it's going to do, it's now going to stimulate aromatase activity. A lot of the testosterone gets converted to estrogen. So the overall testosterone levels are very low, and these may then develop as females or maybe males that are sterile. And then the control female shows you the level of testosterone made in those, which is also very low. So it doesn't take a lot of these compounds to really mess with sexual differentiation in organisms that are using environmental cues as their main mechanism of deciding the sex. So here, just another couple of fun examples to end here. One of these is the organism here known as Bonellia, and this is a marine organism that's shaped sort of like this, where it has kind of a bulbous body and then a long tube that protrudes out and anchors onto the ocean floor. And this large structure is the female, and these eggs, as they develop, if they develop close to the mother, close to this proboscis, the new individual ends up being male. If the new individual gets deposited on the seafloor and is some distance away from the mother's proboscis, it will develop as female. So these are two different scenarios for two different sexes that either close or far from the mother organism, and it's interesting to think about how that might work, and maybe there would be some cues in that female proboscis that were leading to more of a male development. So, but that's also an environmentally based, not genetically based mechanism of doing things. And then fish. Fish have some really crazy abilities when it comes to sexual differentiation. So if you have a male and many females, so one blue guy and several female red guys, male in that population dies, one of the fish, adult fish now, that had developed with a female morphology, can actually go through a shift and change its sex to become a male that can make sperm and participate in fertilization. So these fish have the ability to go down one developmental pathway and then sense their environmental cues, and if needed, certain numbers of them can change to take on the opposite sex. And how could this be done? And it seems to involve neuropeptides. So this is again an endocrine signaling mechanism in the brain of the fish that influences the hormones, and then those influence downstream targets that lead to morphology. And then here's just a spectacular little fish called Prima okinawa that can actually change sex more than one time. So it might initially start out female, and then when the male died, it changed into a male, and then when a larger male came along, it changed back into a female. So a lot of interesting stuff there for sex determination. Any questions on any of this? Alrighty, we are done a bit early, so enjoy the rest of your day.